To understand meiosis, you should at least know the difference between homologous chromosomes and sister chromatids. Homologous chromosomes are two chromosomes in a pair, one inherited from the mother and the other from the father, that have the same set of genes. While the genes are the same on each homologous chromosome, the version of the gene may differ.
Sister chromatids are the halves of the duplicated chromosome after the cell divides, and each homologous chromosome is duplicated before the cell divided. Meiosis is a form of nuclear division that divides one diploid cell, a cell containing two copies of each chromosome, into four haploid cells. There are two rounds of the cell division, meiosis I and meiosis II. Homologous chromosomes divide during meiosis I, and sister chromatids are split and separated into new gametes during meiosis II.
Centrosomes is a small region of the cell that produces protein fibers called microtubules. Before an animal cell divides, the centrosomes, including the centrioles, doubles, and the two new centrosomes move to the opposite ends of the cell. Microtubules grow from each centrosome, forming spindle fibers. The fibers attach to the DNA and assist it to divide between the two cells.
As mentioned before, meiosis occurs, the cell makes a copy of its DNA. When separating the homologous chromosomes, it produces two haploid cells with duplicated chromosomes. Meiosis I happens in four distinct stages, which will be described in chronological order. In prophase I, the first step of meiosis I, the membrane around the nucleus breaks down. The centrosomes and centrioles move to opposite sides of the cell, and the spindle fibers start to assemble. The duplicated chromosomes condense, and the homologous chromosomes begin to pair up. Once paired up, the homologous chromosomes exchange pieces of their chromosomes to create genetic variation, which is called the chromosome crossover. Moving on to the next stage in meiosis I, metaphase I is when the homologous chromosomes pair up randomly line up along the middle of the cell, or the cell equator, by the spindle fibers. The result is that chromosomes, some from the mother and some from the father, are lined up along each side of the cell equator. This arrangement mixes up the chromosomal combinations and helps create and maintain genetic differences. In the third phase of meiosis I, anaphase I, the paired homologous chromosomes separate from each other and move towards the opposite sides of the cell. The sister chromatids remain together, and during this step and throughout meiosis I. The last step of meiosis I, telophase I, is when the membrane around the nucleus forms again, and the spindle fibers break apart, and the cell starts to divide into daughter cells. Ctokinesis is actually the stage where the cell divides into two daughter cells.
Now transitioning into meiosis II, the sister chromatids are separated, which results in cells with chromosomes that are not doubled, and the end result is the four haploid cells. It's important to note that DNA is not copied between meiosis I and meiosis II. Starting with prophase II, the membrane around the nucleus breaks down. Centrosomes and centrioles move to the opposite sides of the cell, and spindle fibers assemble. Metaphase II. Spindle fibers connect the 23 chromosomes at the cell equator. Each chromosome still has its own two sister chromatids during this process. Anaphase II. After the sister chromatids are pulled apart from each other and move to opposite poles of the cell. Telophase II. Last but not least, in meiosis II, the nuclear membrane form around each set of the chromosome at opposite ends of the cell. The spindle fibers break apart and the cell starts to split evenly into daughter cells. Like in meiosis I, cytokinesis is the stage where the cell actually divides.
While mitosis produces two genetically identical daughter cells, meiosis produces cells that are genetically unique from the parent and contain only half as much DNA. Nearly all cells in the body regularly go through mitosis, but some more than others. Meiosis occurs in only plants, animals, and fungi. Mitosis occurs in all organisms except viruses. Mitosis creates a human cell with 23 chromosomes, while meiosis creates a human cell with 46 chromosomes. Mitosis involves one cell division, while meiosis involves two successive cell divisions. In mitosis, crossing over occurs between prophase 1 and metaphase 1, and is the process where two homologous non-sister chromatids pair up with each other and exchange different segments of genetic material to form two recombinant chromosomes, sister chromatids. In meiosis, crossing over is a complex that temporarily forms between homologous chromosomes that is only present in prophase 1, making this the only opportunity the cell has to move DNA segments between the homologous pair.